So welcome everybody to Joseph Crispin Nakoa's Masters of Science thesis defense today. I'd like to tell you a little bit of background about Crispin before he starts his presentation. So prior to starting Hilo as an undergraduate, Crispin graduated from Kamehameha School and was a competitive surfer. And I didn't know about how competitive he was until an article came out for Sea Grant, because he doesn't always share that kind of stuff about himself. I was lucky to first meet Crispin in Mare 350 in 2015. This is our class where we teach the scientific process. And from that point forward, it was obvious his gift for science. His dedication to learning, his inquisitive nature, always wanting to know how to do something better, and his high quality of work. When I had him in chemical oceanography, there wasn't anything that he could not do. After graduating Hilo with a bachelor's in science and marine science in 2016, Crispin participated in the Hilo Pipes Summer Internship Program, where he worked with Luca Mossman at Ho'opono Hale'olono. Following this experience, Crispin went to work for Ocean Era. In spring 2020, when Steve Colbert and I found out that our Hawaii Sea Grant proposal was funded to map out sewage pollution in Hilo, both Crispin and Shayla were the two students who immediately came to mind for the project. And we began to recruit them, and they were really the perfect fit to be able to do that. Today you will hear about Crispin's part of the story, where Shayla presented hers on Tuesday. Crispin was both a Ha'oli Maloa and Hawaii Sea Grant scholar during his time in the Tropical Conservation Biology and Environmental Science graduate program. This spring, Crispin also participated on a NOAA cruise along the west coast of Hawaii Island and assisted the Mo'oli'i community in water quality sampling. We will miss Crispin when he graduates, but we are excited to follow his journey to Arizona State University, where he will be a PhD student working with Dr. Cliff Capono. Today, Crispin will be presenting results from his Masters of Science thesis research. The project that Crispin will be presenting on started from discussions at the Keaukaha Community Association, where community members were concerned about the stink from the Hilo wastewater treatment plant. But we knew that the wastewater treatment plant wasn't the only source of sewage to Hilo's shoreline waters. There are still a lot of homes that use cesspools. So our project that Crispin headed up was to look at sewage pollution along Hilo's shoreline. So to accomplish his goals, Crispin worked closely with the Keaukaha Community Association, the Lelevi Association, and with his fearless team member, Shayla Waikiki. So it's with great pleasure and pride, I give you Crispin Nakoa, who will be speaking about detection of sewage pollution in coastal waters. Sorry, I'm blocking the title here. Waters with river and groundwater inputs in Hilo, Hawaii, USA. So Crispin, please take it away.
All right, aloha, mahalo to everyone for coming to my thesis defense today. Also, thank you for that introduction. That was really cool. It's been a really fun couple of years for me in the TCBES program. As Dr. Wigner mentioned, I got to do a lot of fun activities outside of school, in addition to carrying on my studies and really answering or looking for answers to questions that I've had my whole life growing up here. So thank you to my committee, also Dr. Steve Colbert and Dr. Carla McDermott for all of your assistance in every step of my project. And also thanks to the communities that I grew up in for allowing me to better understand some of the issues we're facing over here. I'd also like to acknowledge all the TCBES faculty for shaping my understanding in the field and also for really shaping me into the scientist I've become. And also to my funding sources, University of Hawaii Sea Grant and Ha'oli Maloa. I couldn't have had the great experience that I've had without your support. Now I'm really excited to get into my findings, but first I'll start with some introductory information. So it's well known that human sewage or sewage pollution is harmful for human health. And especially when it happens in places where we recreate and depend on for resources, it's even more pressing. Sewage has a high bacterial load that's been linked to over 120 million cases of gastrointestinal illness, 50 million cases of respiratory illness, and many cases of skin infections each year. The association between antibiotic-resistant bacteria and human sewage also usually increases the difficulty of treating infections in many cases. Aside from bacterial pathogens, there's other harmful contents like endocrine disruptors, heavy metals, and toxins, which are also present in high concentrations in sewage. And especially in areas heavily impacted by sewage pollution, negative health impacts have been seen. Looking at the environmental side of things, in places where sewage pollution, whether from point source or non-point source sources, eutrophication is commonly observed. Algal blooms usually result, and in many cases, there's following hypoxia. Phase shifts from coral to algal-dominated substrates have been seen and documented in areas with long-term sewage inputs, and just general shifts in benthic community structure. A good example of this is Kaneohebe on Oahu, which was overrun by a species of green algae, and benthic community structures were completely shifted after years of contamination through multiple sewage outfalls within the bay. In the Caribbean, sewage pollution has been seen to, or has actually been linked to whitepox disease, which specifically affects Acropora palmata or elkhorn coral, and it's caused by the colonization of the coral microbiome by the gut bacterium Serratia marcescens. In general, sewage input adds additional stress to coral reef ecosystems that are already facing things from other environmental stressors, such as warming ocean temperatures, coral bleaching, and even direct damage that may occur during storms, ship strikes, or anchor strikes. Recovery of these ecosystems that we depend on so much for both economics as well as a food resource is possible, and it's been seen to take up to decades and even longer, and some changes are thought to be irreversible after point source sewage pollution is corrected. So we have all of these negative impacts of sewage pollution, and the real key to avoiding them and all these other detrimental effects is to properly manage our sewage. We can't help making it, but we can treat it better before we release it into the environment. Pictured here is the general schematic of a municipal sewage treatment plant. I'll refer to it from here on out as an STP. Generally, this would be implemented in urban areas where sewage is transported from homes and businesses to the STP, where it usually goes through at least 2 phases of treatment. In primary treatment here on the left, generally solids will be filtered out and it goes on to secondary treatment, where biological processes will take place and additional solids will be filtered out and turned into a sewage sludge. This is usually disposed of through a landfill, and secondary effluent, which is the majority of what wastewater or sewage is, will be disinfected usually through chlorination and then discharged through injection wells or outfalls. There's also some cases where the effluent may be used for like in places where water is limited and it's recycled. Then in these cases, if there's an industrial or agricultural use, it'll usually also go through a tertiary treatment. But the big take home from this whole diagram is that sewage in STPs goes through many steps and treatments, and the goal is to just reduce nutrients and remove harmful contents as much as possible. Although it is an effective method, STPs are subject to mechanical failure, and there's still a lot of things left over after it goes through this process. It doesn't remove everything that's harmful, but it's considered the best management option currently. In more rural areas, an STP or access to sewer systems might not be available, which leads to the next option, on-site sewage disposal systems. I refer to them as OSDS. There's a variety of types, but the main idea is that they're designed to treat sewage to some degree before it's released back into the environment. So pictured here is the general schematic of a septic system utilizing 2 tanks and a drain field. In this case, raw sewage would be transported from a house, a few houses, or even businesses in some areas, and it goes into the tank and it's allowed to settle. Solids will sink to the bottom, a scum layer will develop on the surface, and the majority of it would actually be effluent with dissolved particles and small particulate matter. As it settles out, it goes into the next tank where the process repeats itself. And the real key to a septic system or more modern types of OSDS is the drain field. This is specifically designed to slow the flow of effluent before as it's being released into the environment, so biological processes can take place and treat it, remove nutrients, and remove harmful contents. There is a downfall for this, the few downfalls for this, which would be that They can't be located in close proximity to surface waters, and they need to be a set distance above water table. So they're not applicable in every area. And another thing is that they need to be properly maintained. If the tanks fill up, it generally will decrease the ability of the drain field and the tank system to slow the flow of sewage and allow for biological processes to take place. Finally, the last type of OSDS I'd like to point out was once a really common method of sewage disposal. It's a schematic. Here's a schematic of a cesspool. This is now considered an antiquated method for mainly the reason that's pictured here. It's basically where sewage would come from a house and be piped into a hole in the ground in close proximity to the house. This hole is usually lined or can be unlined or lined, but it's usually with an open bottom. And when there are unsaturated sediments, it does remove some nutrients and contaminants, but these are often long-lasting systems that saturate soils around them. If not, they're also usually located close to the water table, and there's not really a lot of soil in between the water and the sewage in the cesspool. So from there, it will usually contaminate groundwater, where it can impact drinking water wells, municipal wells, move through groundwater through rivers and streams to the shoreline, or go all the way out to submarine in the groundwater through submarine discharge. And we're adding contaminants and excess nutrients where we see these effects in the nearshore waters. Hawaii has a cesspool and even an OSDS problem. We're the last place to heavily rely on the use of OSDS, especially at the scale that we do. In total, there's over 110,000 OSDS here. As you can see in the figure, they're illustrated by the light yellow points. And one thing I'd like you to notice is that there are some clusters here and there, but for the most part, they're all located around the coastline, which is related to the way that humans usually develop near the coast first. And that's where we like to live. It's nice. It's close access to everything. It's like nicer terrain, flatter in most cases. But the problem with that is that we have almost 60,000 OSDS on Hawaii Island, between 15 and 20,000 on Maui, Oahu, and Kauai, and another 2,000 OSDS on Molokai. Of these, about 80% are cesspools. And with the known effect of cesspools that I described in the previous slide, and these cesspools being located in coastal areas, we can see where the problem might come from. To address this, the state of Hawaii recently set the goal of eliminating all cesspools by the year 2050, mostly trying to replace them with other types of OSDS that are more modern. And this was done through the passing of Act 125 in 2017. This is kind of in the early stages still. It was just a few years ago where this was established, and the state's made a few attempts to overcome the difficulty of reaching this huge goal. The main thing that's been done was the establishment of a cesspool conversion working group, which is tasked with identifying priority areas where we should focus our resources first, studying other areas that have already dealt with this, since Hawaii is the last place to need to really go through cesspool removal, and also to just better understand different options that we have to improve the feasibility of reaching this goal. A little bit more on the problem that we have. Dye tracer tests have confirmed that OSDS are connected to shoreline waters, and that sewage travel time can be from hours to days in some cases, especially in coastal areas. Studies that were specifically done in Pu'uko and West Hawaii by Wiegner et al. Have seen that hydrogeology rather than OSDS type most influence the sewage travel time. And if we're trying to convert all cesspools to more modern types of OSDS, this is something that needs to be looked at. It might not really be applicable in some areas, especially if there's a really permeable substrate. Many areas would actually benefit from the installation of new STP or expanding existing sewer systems to connect to STP that are already there. And even if this was possible, there's still many homes in really remote locations where it simply couldn't be done. And if it was done, it would come at a very high cost to the state, and it's unlikely that we'll be able to accomplish this everywhere it needs to be done within the 30-year timeline that we have left. Currently, there's a few incentives for homeowners that utilize cesspools. There's low interest or no interest loans, and there's also previously a tax credit. But overall, the high cost has really deterred homeowners from converting their cesspools to other types of septic systems. And it's really quite an investment. It can cost upwards of $20,000 and up. And to better address this in the future, there will likely be more incentives as the state identifies and finalizes their priority areas for cesspool conversion. So now we move on to Hilo, Hawaii, a place that most of us are familiar with. We have nearly 11,000 cesspools in Hawaii, or in Hilo, in the district. In the Hilo Bay area, which is mostly pictured on this map, there's about 8,700. And yellow points on this map indicate cesspools and other types of OSDS, but again, remember, they're mostly all cesspools. Also on this map are both perennial and ephemeral rivers and streams, which are light blue lines. And as we can see, a lot of the OSDS in this area are either located along the coastline or clustered adjacent to rivers that can transport sewage to the shoreline. Hilo is also the highest freshwater discharge area in the state, which is estimated to be 2.5 million cubic meters per day from groundwater and 1.2 million cubic meters per day from rivers. This was an estimation made in the 1980s, and it's also reflective of ambient conditions. As we know, in Hilo, we're on the windward facing side of the island. We experience a lot of heavy rain. We also get frequent storms during hurricane season. And this discharge is actually increased in many periods, many points of time. But anyway, the freshwater all the freshwater present here really illustrates the likelihood that all of these OSDS could be contaminating groundwater and streams and getting to the shoreline. In addition to the number and density of cesspools in Hilo, there's also a sewage outfall, which can be seen by the brown cross over here. It is from the Hilo STP that treats sewage through fixed film secondary treatment. And since its construction, community members have voiced concerns over environmental changes in the area, but they haven't been well studied yet. Another factor that we have here is this about 3-kilometer breakwater, which protects Hilo, Port of Hilo, and Hilo Harbor, but it also has been documented to reduce ocean mixing and traps pollutants from groundwater and 2 rivers that flow into this area. Wailoa River is fed by ephemeral streams and also is one of the highest groundwater discharge points in the state. The Wailuku River in this area is actually the basin for the largest watershed in the state. And both of these flow into the enclosed portion of the bay, obviously giving us some concern. Despite all these risk factors present in Hilo, there's only been a few studies of sewage pollution in Hilo Bay, mainly focusing within the breakwater. 2 studies by Wigner et al. Have looked at the input of sewage from those two rivers, Wailoa and Wailuku, in addition to a few groundwater stations. But the main focus was to find differences under low and high river flow conditions at the shoreline, in rivers and streams, and throughout the bay. Fecal indicator bacteria, which I'll describe a little better later, or FIB, were present in streams and groundwater. 3 more recent studies by Economy et al. And Gherkin et al. In 2019, 2021, and a current article that will be published soon, all looked at the possibility of having environmental sources of these FIB that are known to be found in streams and shoreline waters and Hilo Bay. Economy et al. Found that FIB were present in soils in addition to streams, the bay, and groundwater. Gherkin found that FIB were present in soils, streams, and beach sediments. And also antibiotic-resistant bacteria were present in these areas as well. So what that really looked at was trying to identify if the FIB that we're measuring really come from sewage, or they're accumulating in the sediments or somewhere else and then being flushed out during these heavy rain periods. In addition to these scientific studies, the Hawaii Department of Health has a beach monitoring program that is throughout the entire state. In Hilo, it monitors 5 beaches for concentrations of FIB, and it uses this data to assess the safety of our recreational marine waters. And they also close beaches when needed. Some of you may have seen the signs posted, beach closed, bacteria levels elevated, and they'll do follow-up testing if that is the case. As most of us also know, we have more than 5 beaches in Hilo. So the Surfrider Foundation previously monitored an additional 7 beaches. I think they may have had 2 stations that were overlapped, but the idea was to get a better understanding at a broader scale. Since the beginning of our study, this program was no longer active, and that kind of gets into our goal. So we had 3 main goals, which included a larger section of the coast than has previously been studied in Hilo. The first objective was to determine if HDOH water quality standards were exceeded for FIB, nutrients, chlorophyll A, and turbidity. And all these standards were set by the HDOH or recommended to and acknowledged by the HDOH. The second objective was to examine the relative importance of groundwater and rivers as sewage pollution vectors. And the third objective was to examine the efficacy of sewage indicators in a tropical setting with many freshwater inputs. The study site included 20 stations, which spanned 5 ahupua'a. To describe them a little bit, beginning in the north, there's the Kaiviki and Alai ahupua'a. We had 2 stations there. Pu'ueo and Pi'ihonoa ahupua'a were divided by Station 18, the river, or Wailuku River, which was Station 18. And then the rest of our stations fell within the north-facing portion of the Waiakea ahupua'a. These stations included all the beach parks in the area, 3 river mouths, and other areas of community importance, as you might notice a few clusters of stations in certain areas. They were all sampled from July 2020 to October 2021 at low tide in the mornings, because that's believed to be the most indicative of fecal indicator bacteria before daylight influences them. And also they were sampled on 2 consecutive days each month, 10 stations per day. The coastal waters at many of these stations, really all of these stations, are frequently utilized by the community and visitors for things like surfing, fishing, and cultural practices, myself included in that group. And it also illustrates the importance of identifying sewage pollution and really better characterizing water quality here at the scale that we've done it. So the 20 stations were put into 3 groups based on physical attributes and salinity data that were collected throughout the study period. As you've seen in that short video, that's what most of our stations look like. There was a large gushing groundwater spring. And stations that didn't have a spring like that were in close proximity to river mouths. So to better look at the influence of sewage vectors, we grouped them by river influence stations in the RI group, which were at a river mouth or in the sediment plume of river mouths. These stations were Wailoa, Bayfront, Wailuku, Honoli'i, and Honoli'i Stream. In the high groundwater influence group, or HGW group, they were characterized by a visible spring and an average salinity less than 10. These stations were Lele'iwi, Waiolena, Lalakea, Carlsmith, Haleolono, Chalks, 2 stations at Puhi Bay, 1 near the ocean and 1 more by the road in the protected area, and also Reeds Bay. Stations that had a low groundwater influence were put into the LGW group, and they fell into this category if they did have a visible spring, but their average salinity was greater than 10. These stations were Lehia, also known as King's Landing, Ka'ilivai, also known as Richardson's, near the Hilo Yacht Club, at Onekahakaha, at Puhi Bay, near where the outfall goes offshore, and at Liliuokalani Gardens. To sum it up, there were 5 RI stations, 9 HGW stations, and 6 LGW stations. This study used a multi-indicator approach that was developed by Abaya et al. In 2018 in her study of sewage pollution in Pu'oko, and also used her sewage pollution scoring tool, which incorporated many of these indicators to gain a more comprehensive understanding of the level of sewage that's present. The 3 FIB measured were Enterococcus species using the IDEX method. Pictured here is a picture of a positive IDEX tray, or positive Enterococcus reading using an IDEX tray. Illuminated cells mean that Enterococcus is present, and non-illuminated cells mean that there was no Enterococcus present. And a concentration is calculated by counting the large and small wells, and you get a value that is most probable number per 100 milliliters. We also measured Clostridium perfringens using membrane filtration and MCP plating. And finally, for FIB, we also measured human-associated bacteroides using the HF183 quantitative polymerase chain reaction, or qPCR assay. Nutrients measured were nitrite plus nitrate, ammonium, total dissolved nitrogen, phosphate, total dissolved phosphorus, and silicic acid. Macroalgal tissues were assessed for the delta 15N and percent nitrogen content. Nitrate and water samples were analyzed for the delta 15N and delta 18O to be used in our mixing models. Chlorophyll and turbidity were also measured for their known association with sewage pollution. And when possible, lab analyses were conducted by myself or with my team. And if special instruments were needed, they were sent off to the Hilo Analytical Lab for nutrients and stable isotope data. And also human-associated bacteroides were sent to the McClellan Lab at the University of Wisconsin. So getting into results of my first goal, many of the sewage indicators measured did have water quality standards. This table is illustrating these comparisons with the freshwater influence group here on the left, the indicator on the top, median and range of each station's value for each indicator is represented as the numbers. The bolded values with shaded cells indicate exceedance of the standard, which is listed below the column for each indicator. Some things I'd like you to notice are first, that these standards are for estuaries and also embayments under wet conditions. They're all the same between these two environmental areas, but chlorophyll A actually was 1.5 for embayments under wet conditions, and estuaries were 2 micrograms per liter. So looking at our comparisons, Enterococcus concentrations, chlorophyll A, and turbidity were all exceeded by the river group alone. All freshwater influence groups exceeded nutrient standards. And previously measured FIB concentrations were actually a lot higher than we saw in this study, especially for Clostridium perfringens, which was actually mostly absent. But it's been documented from 0 to 45 CFU per 100 milliliter in Hilo, and even higher in other parts of the world experiencing sewage pollution. Not pictured in this table is the absence of human-associated bacteroides, which was also previously documented, but was absent from all shoreline stations throughout the entire study period. Nutrient concentrations we observed were much higher than past studies of Hilo Bay, but still lower than other areas of known sewage pollution with well-documented problems, such as Maui. Chlorophyll A and turbidity standards were seen to also be previously exceeded much more by other rivers and streams in Hawaii, as well as Hilo Bay, with standards being exceeded by as much as 300 and 400%, as opposed to the 25% they were exceeded in our study. In addition to measuring all of these sewage indicators, we also looked at OSDS density, doing a little bit of geospatial analysis. OSDS density within a 1 square kilometer area was calculated by creating a buffer around station points, which can be seen as the labeled station point, the light colored outline of the buffer, and OSDS within this buffer are indicated by bright yellow, and then that same yellow that we've seen in previous figures was indicating OSDS beyond the buffer. The EPA suggests that areas with more than 15 OSDS per square kilometer is likely to experience groundwater contamination, and this density was exceeded in all 3 of our groups by as much as 187%. The highest OSDS density we saw were in the RI group, mainly coming from stations 18, 19, and 20. And then it was followed by the HGW group, which isn't as easy to point out because they were mixed spatially, but the LG group actually had the lowest number of exceedances. And if you think of this ordering, RI group, HGW group, then LGW group, it's kind of in the same order that the exceedances of water quality standards were seen. The RI group exceeded almost everything. The HGW group was next in nutrients only, and then the LGW group usually had lower values in nutrients only. 13 of the 20 stations, when we looked at the data as a whole, exceeded this recommendation. And looking at some correlations between this value and other indicators, there were weak positive correlations between OSDS density and C. perfringens and all nutrient measurements of nitrogen and phosphorus. There were also weak negative correlations with ammonium, chlorophyll A, and turbidity, which is kind of interesting because they're usually associated with contaminated waters. Moving into my second goal, rivers are known to harbor sewage-associated bacteria and viruses, increasing the risk of contracting illness and infection. They also have elevated concentrations of nutrients, mainly ammonium, nitrogen, phosphate, and silica in comparison to coastal waters. The figures here illustrate the FIB and delta 15N values, which are all highest in the RI group, with freshwater influence groups being shown on the x-axis, measurements or values being shown on the y-axis, and statistical results displayed on each figure. Also note that A indicates the highest grouping and different letters indicate different groupings. So in addition to these indicators that are shown on this slide, ammonium, chlorophyll A, and turbidity were also highest in the RI group, and these results agree with others that identified rivers as major sources of sewage pollution to shoreline waters. One thing I would like to point out is that there were times where FIB and the delta 15 and the macroalgae were actually higher at groundwater groups, but on average, the river stations were the highest. Looking more at water or groundwater, as we already discussed, there's a growing concern that it may be contaminated by OSDS, especially where there's large volumes of groundwater and widespread distribution, as well as high densities of OSDS. It's even more relevant in places like Hawaii, where we have very permeable basalt substrate that could allow for the really quick transport of pollutants from an area to the shoreline where we recreate and depend on. Many other areas, including parts of Hawai'i Island, other Hawaiian islands, in California and in Mexico, have all found groundwater to contribute high concentrations of both nutrients and FIB to the shoreline. And it's frequently observed to see delta 15N macroalgae values that are elevated in close proximity to shoreline springs in these areas. The figures here display the same information as the previous slide, but this time for indicator measurements that were highest in the HGW, or in the HGW group. These are nitrite plus nitrate, phosphate, phosphate and silicic acid. In addition to these ones also all being highest in the high groundwater group, we also saw total dissolved nitrogen, total dissolved phosphorus, and percent nitrogen content in macroalgae were also highest in the HGW group. Although the LGW group didn't have the highest value for any of our measurements, both groundwater groups were characterized by low FIB concentrations and high nutrient concentrations, a finding which suggests that sewage is present in groundwater, but it's very diluted, and the nutrients that we're seeing may actually be from non-sewage sources. So as we saw, there is contrast between low FIB and high nutrients, which are somewhat contradictory of each other. And to address this, we use that sewage pollution scoring tool that I described earlier. To better explain how the tool works, it's basically this equation. Each indicator is put into a level based on its exceedance or falling its level in respect to its respective standard. There's also a weight depending on the, indicative nature of the measurement to sewage pollution. So more indicative of sewage pollution would be given a weight of 3, whereas nutrients can come from many sources, they're given a weight of 1. Total scores are summed from by filling out this equation and adding everything together for each station on each sampling date. And they're placed into categories from low impact from 11 to 17, medium impact from 18 to 25, and high impact from 26 to 33. In this study, it can be seen on this figure that all values, or all freshwater influence groups, had a value of 17, and they were similar among groups, indicated by this p-value being greater than 0.05 over here. All stations did fall right at the high end of the range for low-level sewage pollution, but also when looking at individual stations throughout the study period, there are many occasions where stations fell within the medium category, and every station was in this category at least once. So all of those previous measurements that I was presenting to you made an attempt to quantify the level of pollution, but it didn't really do anything to address the possible sources of pollution. We can make inferences, but this stable isotope mixing model method has actually been used to estimate the percent contribution of nutrients to an area in many other studies. To better display the contributions of nitrate sources, the results of our isotope mixing models have been shown here and on the next 2 slides, grouped by station, with colors of boxes representing the group that the station was in. Boxes also represent Bayesian 50% credibility intervals, and the proportion contribution is on the y-axis. Oh. Sorry about that. So here we can see the contribution of groundwater to each station, and we can see that it's highest in the river group, as high as 98%. Most stations were groundwater contributed 60 to 80% of nitrate, and the high contributions of groundwater to the RI group may be due to the base flow of all rivers actually being comprised of groundwater. One thing I'd like to point out and keep in mind for the next few slides is that Station 9 had the lowest contribution of groundwater. Moving on, we can see the contributions of the STP outfall range from 14 to 40%. It was highest at Station 9 at 40%, but most stations were actually within the range from 14 to 25. Station 9 is an area with a small breakwater, and it's a small beach with a small breakwater with a little groundwater influence, as it indicated by the low groundwater grouping. And we're thinking that this may suggest that pollutants could be coming in at high tide, but the reduction of ocean mixing water and the lack of groundwater to dilute what's there is actually leading to an accumulation of nitrate and inferring that it also is accumulating pollutants in this area. To explain this blank area from station 14 through 20, these were stations that were spatially separated from the STP outfall, and they were excluded as potential sources to these stations. Finally, we looked at sewage from OSDS and found it contributed the smallest proportion of nitrate to stations. The highest contribution was here at station 18, that was 16%, and most other stations fell within the range of 1 to 9%. There's a few possible explanations for this low contribution. One being that there's little sewage present and that there's a lot of groundwater diluting these. The other option is that OSDS source data we obtained weren't from areas in Hilo, but they were of areas on other parts of Hawaii Island. And they were assumed to be similar to OSDS in Hilo. But when we were examining preliminary isoplots that were created to identify potential sources to our stations, the OSDS data that we had showed a larger distance than expected between that and effluent from the wastewater treatment plant. But it still was included in the model because there were some stations with data points in this range. But for a future improvement of this type of study, we could possibly gain data from OSDS from Hilo specifically. Finally, moving into my last goal, we wanted to better understand the associations and how to interpret different sewage indicators. So we used Spearman rank correlation test to determine significant correlations in their direction. Here on this figure, it's a little complex, but I'll break it down in a minute. But significant correlations are indicated by these asterisks. Positive correlations are indicated by red tiles, negative correlations by blue tiles. More pronounced and darker colors are indicative of stronger correlations. So some things I'd like to highlight are that FIB were positively correlated with each other and also chlorophyll A. Clostridium perfringens was positively correlated with ammonium, and Enterococcus is also positively correlated with delta 15N in macroalgae. And although these correlations are weak, this suggests that they may be the most indicative of sewage here, as ammonium is often the most common form of nitrogen in sewage, and positive correlations between both chlorophyll A and delta 15N of nitrate are both seen to be significant, kind of agreeing with this conclusion. Another thing I'd like to highlight is that FIB were not correlated to other nutrients, and neither were chlorophyll A and delta 15N in macroalgae were mostly negatively correlated to other nutrients. So what we're thinking is that it's possible that the high nutrient concentrations we've been seeing at the shoreline are actually from a source other than sewage. To summarize this figure, it seems that in a tropical setting with high inputs of fresh water, that FIB, ammonium, and chlorophyll A can be most indicative of sewage, and that the high amount of nutrients we observed at our shoreline stations likely come from sources other than sewage. Using multiple sewage indicators allowed for a better assessment of coastal water quality than just relying on a few sewage indicators, as has been done in some previous studies, as well as is often used by monitoring agencies. This is exemplified in the study by extremely high nutrient concentrations, suggesting a major issue that needs to be dealt with right now. But then we accounted for FIB concentrations and lowered delta 15N values in macroalgae, suggesting that there is a problem, but it's not like an immediate threat that needs to be dealt with right now and don't go in the water. Incorporating all of our sewage indicators into a sewage pollution scoring tool allowed us to conclude Hilo is impacted by low-level sewage pollution, but it's also important to remember that this had an extended sampling period over 17 months. And although sewage pollution overall was in the low category, there are many times where coastal waters were impacted by medium-level sewage pollution and likely presented a greater risk to swimmers and the environment at these times. Water quality standards for nutrients are regularly exceeded throughout Hilo. Standards for FIB, chlorophyll A, and turbidity are occasionally exceeded in Hilo's coastal waters, but most commonly in areas influenced by rivers. When using common sewage indicators, they need to be assessed with an area-based understanding of the environmental factors present to illustrate the extent of sewage pollution there. Dilution, as we saw, can make the detection of sewage pollution difficult, especially when there's high groundwater and river inputs. The results of this study will provide insight for decision-making and hopefully will be included in upcoming discussions of sewage management, as well as cesspool conversion in Hawaii and other areas utilizing OSDS and cesspools. Future studies of sewage pollution should also include a variety of indicators to really gain a full understanding of the effects of sewage pollution on water quality, as well as the influence of different vectors of sewage pollution on water quality. Mahalo again to everyone that's made this work possible, especially my advisor and my graduate committee. I'd also like to shout out Shayla Waikiki and congratulate her on successfully defending her thesis earlier this week. We both worked really closely with each other on each other's projects, and I couldn't have done it without her. She's pictured here in the center. Also, Nick Story, who is taking this picture. Thank you for the lab assistance and field assistance, as well as handling the DNA processing. Other people who have helped a lot are with lab and fieldwork are Finn Real and Walter Bolger. Mahalo, you guys. Always been there for when I needed last minute help on my sampling days. I believe Walter's here today. And also thank you to Devin Aguiar. He was just finishing, he just graduated the TCBES program when we started, and he was conducting a similar study on benthic water quality in Pu'uko. And he really helped us get into the swing of sampling, helped us plan our days, went over SOPs with us, and kind of just really let us know what to expect. Finally, again, thanks to my funding sources, Ha'oli Maloa and the University of Hawaii at Sea Grant. I really did have a great time in this program, and I couldn't have had this as good of a time as I did in graduate school without your support. Also, thank all of you for attending my thesis today, and I can take a few questions now before I go on to meet with my committee. Mahalo.